All right, when I last left you, I put this question up. Sickle cell disease is caused by mutation of the beta hemoglobin gene that changes a charged amino acid, glutamic acid, to valine, a hydrophobic amino acid. Where would you expect to find glutamic acid? Well, glutamic acid is, as we see here, let's find it, is charged. It has a charge to it. Therefore, it would be hydrophilic. Therefore, it could be on the exterior surface of a folded protein. So it would be on the outer side. Now, when you turn it to valine, that valine is going to fold away from water and completely change the shape of that protein. Like a piece of paper crumpling up in a different way. It'll fold differently, changing the shape of that protein. The last molecule in this chapter is the nucleic acids, and I'm not going to hit it very hard because we have a whole chapter on this, right? But DNA and RNA. And DNA and RNA code ultimately for the shape of proteins. And so DNA is found in your nucleus. They are polymers of nucleotides, so their monomers are called nucleotides. You glue them together to make the polymer nucleic acids. And the major role are, in this case, are information storage, but of course nucleic acids, or at least modified nucleotides, are also used for energy in the form of ATP. These have the basic structure of a sugar, a 5-carbon sugar, a pentose, with a nitrogenous base stuck on one side here to the first carbon, and then on the 5th carbon up here we have a phosphate group attached. You glue a bunch of these together through a covalent bond, and you get what is known as a phosphodiester bond. This occurs when DNA replicates. Phosphodiester bonds, that's the covalent bond here in the backbone. And we can form a backbone of DNA. And then if we form another backbone over here running the opposite way, hydrogen bonds will form between these nitrogenous bases, forming that double helix that I'm sure you're familiar with. So the bases vary. There are pyrimidines, which have 1 ring, purines, which have 2 rings. The purines are A and G, adenine and guanine, while the pyrimidines are C and T in DNA, but U instead of T in RNA. The sugars are about the same in DNA and RNA, but the 2nd carbon here in DNA has an H instead of an OH, making the sugar deoxyribose. It's missing a sugar, versus ribose in RNA. So if you see a molecule like this and it has, here's the sugar, if it has an OH here, you know it is RNA, and if it has uracil U attached to it, you know it's RNA. RNA is typically a single strand, whereas DNA is usually a double-stranded molecule as well. The polymers have a little bit different structure. So overall, what we've seen in this chapter are that sugars, monosaccharides and polysaccharides, are used for structure in the case of cellulose, as well as energy storage. We've seen lipids, which are used for energy storage, as well as communication. And if you've learned about membranes, they also make phospholipids. They are part of a membrane. Proteins have so many functions, we're not going to list them here, but communication, enzymes, structure, etc. And lastly, nucleic acids are information storage molecules that allow us to pass genes on from one generation to the next, and they code for proteins. They also are involved in energy in the form of adenosine triphosphate, or ATP, the molecule that powers almost all of the processes that keep you alive. All right, so review that chapter. It'll go into a little more detail than I do. Check out guided reading questions and all the available homework, and you will knock this out of the park.